Hi, Mike. I believe we can start already, at least to welcome everybody who is already in this webinar. Today's webinar is about the USP 922 water activity new chapter. First, we would like to give you the welcome and guide you a bit through the different functions and things that we would do during this webinar. My name is Paula Braco. I'm a study manager at Lighthouse Instruments for 3 years now. I work at the office here in Amsterdam, in the Netherlands. Here we also support our customers in Europe with measurement services, but also with their own instruments on site. Yeah, my main role is to manage a portfolio of customers' projects here in Europe and collaborate also for R&D projects, specifically in moisture application, and therefore my interest also in the water activity application from Lighthouse side. If you go into the bottom part of your screen, you will see different functions from Zoom. The most relevant one is the Q&A button. If you click there, you can actually place questions, and those will be answered by the panelists present in this webinar. There's also a chat function that you can use, but we encourage you to place questions already during the presentation. But Mike will take some minutes at the end also to take some questions and discuss them with you. Also, I would like to mention that this webinar will be recorded. So we hope to send you a link to the recordings after this session, so you also have the chance to see it again or share it with your colleagues. Yeah, we had almost 400 people register to this webinar. We are still seeing the numbers increase while I'm speaking, so I think this will still happen. But yeah, Mike, I think I can give you now the screen and you can start with your presentation.
Thank you so much, Paula. My name is Mike Laly. I'm the Validation and Application Specialist for Lighthouse in the United States. I work from our Charlottesville, Virginia office, and my role is primarily to help with methods development, methods validation, and on-site work with clients. I'm a chemical engineer by training. Been with Lighthouse for 10 years and have worked on the vendor side with the pharmaceutical industry for about 30. So I'd like to just start. We're going to be talking about USP 922, the new chapter that defines water activity. Chapter was published in USP NF 2021 edition 1 on November the 1st, and the chapter will become fully effective on May the 1st of this year. So in today's webinar, we're going to talk a little bit about what is water activity, how does it impact critical quality attributes, how does water activity change over time in your packaged product, where is water activity data used during stability protocols, and how does Lighthouse measure water activity? So as you can see, there's 5 sections. I'll present some information, some data, and approximately at the end of each session, I'll have a polling question. As of right now, we've got 126 people participating. And I think all of us would really appreciate it if we can share some information about what we're doing in our own organizations and best practices, and it will help all of us as we implement this new chapter this year. So first, what is water activity? It is a measurement of the equilibrium concentration of water in the air above your tablets or capsules, divided by the concentration of water that would be there if the sample was pure water. So there's a little illustration there. I think we all understand that if I put a pill inside of a sealed vessel, I'm going to slowly desorb water from the solid. It's going to enter the headspace, and it will reach equilibrium where water molecules are both entering and then reabsorbing. this measurement is really a measurement of the chemical potential of the drug product, of water that is available to participate in undesirable chemical, physical, or biological reactions. It's very much aligned with Gibbs free energy. It's really showing how water is being held to a surface or to itself that's been absorbed to a surface. And it is also equal to the relative humidity inside your package divided by 100, as long as that's done at the exact same equilibrium and temperature as the measurement. So just want to spend a minute and talk about some common excipients for oral solid dosage products. First, we'll pick a salt, dicalcium phosphate dihydrate. So as I think we all recognize, this water is going to be held inside the crystalline lattice of the salt itself. If I do Carl Fischer or loss on drying, I would see that DCP dihydrate is 21% by weight water, but because it's held in the crystalline lattice, if I measure the water activity, it's only 0.17. The water is very tightly bound in the crystal. We then have a material like pre-gelatinized starch. That's a very amorphous material that's been treated with temperature and water. It has a water content by Carl Fischer of 8% and a water activity of 0.3. The water in this case is typically a monolayer loosely absorbed to the starch molecules themselves. And then finally, I'll pick a material, an excipient like monocrystalline cellulose. These materials have both crystalline and disordered states or portions inside the structure. The amount of water is really quite low, 4% by weight, but it is very, very available. Its water activity is 0.46. It really exists as multi-layers with significant amount of free water. So these are what your individual components would look like if you were to open the drum or assay the raw materials before they enter the process. As soon as these materials are mixed, things begin to happen, and water will move between them to reach equilibrium. And what is always true is water moves from high water activity to low water activity. So even though dicalcium phosphate dihydrates 21% by weight water and MCC is only 4%, the difference in water activity predicts, and it is in fact true, that water will move from monocrystalline cellulose to the salt until all three components come into equilibrium. So water activity is changing constantly during your manufacturing process, during storage, and during use. So I think it's useful to take a look at how does water activity change. So in this slide, we're just going to take 100 milligrams of three different ingredients, and we're going to first precondition them at 75% relative humidity until they reach equilibrium. And then we're going to measure the water content, and that's times 0. And then we're going to equilibrate them at 75% relative humidity, and then measure the water content again and report the change. So the amorphous pre-gelatinized starch started 100 milligrams, contained 7.1 milligrams of water. It went to 15.7 milligrams, a change of 8.6, and in a percentage from its initial 122% change. The monocrystalline cellulose under these changing conditions was 141% change in water weight. And the active pharmaceutical ingredient, which is a crystalline drug, very dry, had come from a crystallization process and had been dried. Water content was only 0.2 milligrams per 100 milligrams of drug. After exposing it to 75% relative humidity, the API contained 1.4 milligrams of water. So weight change of 1.2 milligrams, and percentage-wise, that's a change of 486%. It's not a lot of water weight, but because it's a crystalline material, this water is on the surface and absorbs very quickly. So let's ask the same question, but let's have these materials that were conditioned at 25% relative humidity. Now let's just add 1 milligram of water to each of these preconditioned samples. So you can see it's the same question, how do things change? But here we want to look at how do they change when I'm changing the percent water, right? So 1 milligram of water is changing each material by 1% by weight. So they all started at 0.25 water activity. That's equal to a relative humidity of 25%. For the pre-gelatinized starch, if I add 1 milligram, the water activity goes to 0.37. That's a change of 48% in water activity. The monocrystalline cellulose goes from 0.25 to 0.42. That's a 68% change. And the active pharmaceutical ingredient goes from 0.25 all the way to 0.71. So change of 0.46 units, 184%. So many of us have worked with water absorption isotherms. And you may recognize as what we've looked at this data as individual points, we could actually measure at multiple water activities, measure the water content, and then plot that data. And what we'll see is percent water, the vertical axis, versus water activity is very unique for each individual ingredient in your formulation. You'll also see that this relationship is non-linear. And by that, in part, we mean that the slope, the change in water weight divided by the water activity, is different at each of the different points along this path. It's a non-linear relationship. Each ingredient has its own unique absorption isotherm, and mathematically, this is normally expressed with the GAB function, the Guggenheim-Anderson-de Boer equation that can mathematically represent this continuous non-linear change in water activity versus water content across the range of water activities that we would expect to find in a pharmaceutical product. So just to summarize this section, total water content is measured by Carl Fisher. All of you have been doing this for years. It's USP 921, is very different than water activity as defined in USP 922. Carl Fisher and water activity are different things. Each ingredient in your formulation has its own unique water activity versus water concentration, and this relationship is typically plotted as an absorption isotherm. Even small changes in the formulation, shifting the concentration of monocrystalline cellulose versus pre-gelatinized starch, adding a super disintegrant, changing a drying process, changing a wet milling process, even very minor changes in your formulation and your manufacturing process can have a very dramatic impact on water activity. And as we'll see in the next section, the reason that this is important is water activity measures the amount of loosely bound water that is available to participate in undesirable chemical, physical, and microbiological reactions. So that's the end of section 1. I'd love to pop up a polling question. So I'm going to read this. There's now 152 of us online. I'm going to ask you to try to respond relatively quickly, allow you to vote or answer kind of this question. it's really to get a sense of where each of you are at in the process. When will you implement water activity testing? Maybe some of you've already fully implemented it. Maybe some of you hope to implement it by its official date, May the 1st. Some of you may be looking at extra time because of budget or other timelines and hope to implement by the end of the year. Some of you, I'm sure this is a brand new topic, and what you're doing is you're considering implementation. And for some of you, just because of the work that you do or the types of compounds that you're working on, maybe you have no plans for water activity testing in the future. So I'm going to give everybody just a few more seconds and we'll kind of see what this looks like. Well, people are still answering, so I'll cut it off at 1 minute. Wow, pretty good response rate. Thank you everybody for getting in there. It looks like we'll get to about 100 people responded here in the first minute. 32% are already doing it, 25% have no plans, and everywhere else is somewhere in between those timelines. Thank you. And there's the results. So, moving on. What critical quality attributes are impacted by water activity? So each of these areas is defined in USP 922, and there's ample literature available to show the effect of water activity on these, and we'll be going through some examples in the next slides. If water activity increases above critical level, you can begin to see growth of objectionable organisms on your non-sterile products. As water activity increases, you can begin to see an exponential increase in the degradation rate of moisture-sensitive active pharmaceutical ingredients. For some of you, it may be new to hear that changes in water activity can have a significant impact on disintegration and dissolution rates and cause product to fail on stability. And I think all of us that have worked with these areas have seen that water activity can have a pretty significant impact on physical properties, including hardness, friability, and swelling. So what I'd like to do is give an example of each. Lighthouse has application notes available on the website. We have a whole section for water activity, and you'll find much more in-depth discussion of these in our application notes. So once water activity goes above 0.75, if the humidity inside your package is above 75%, harmful microorganisms can begin to multiply. Below 0.75, you may have organisms in your formulation, but they are not multiplying. That is, unless they are molds or funguses. Molds and fungus can grow at a lower water activity, as low as 0.61. And part of the reason that this is important is for many years, USP Chapter 1112 has allowed pharmaceutical companies to use water activity testing in lieu of microbiological assays during the stability protocols. So Chapter 1112 has allowed you to do this. Now that 922 has been published, you have a clear path and understanding of how to do the water activity testing in lieu of microbiology. I'd like to give an example on degradation. This is a moisture-sensitive compound. It's a paper published by Gettin and Kesslering in 1977. The effect of temperature and relative humidity on diazepam stability. They published an accelerated stability protocol using temperatures between 40 and 80 degrees centigrade. And just what I want to show here, the chart graph on the left is showing the degradation rate in mass units per day. And you'll see that these are a very non-linear type of relationship expressed against humidity for the different temperatures that were studied. If we take the log of that degradation rate, do a semi-log plot by having the log on the vertical axis and humidity on the horizontal axis, you'll see that I now get a straight line. This is showing that I have a exponential function, and at 42 degrees centigrade, the temperature that's closest to the temperature that your product might see, the degradation rate increases by 75 times, 75-fold, when the water activity moves from 0.4 to 0.7. So this is a dramatic and clear example of how large of an impact water activity can have on a moisture-sensitive active pharmaceutical ingredient. In this application, just summary, want to talk a little bit about dissolution time. This is in a paper from Vidal, effect of accelerated aging on dissolution of ciprofloxacin. This active ingredient is not itself moisture sensitive. The study had 14 different formulations of this one active. Formulation number 6, which is asterisk there, is the reference formulation. It's actually a formulation they made in their lab. All the other formulations were commercially available product, or many of them were commercially available product. So what they did is they took samples from each of the different formulations and tested their dissolution time as initially received, and then after accelerated aging conditions being 40 degrees centigrade and 75% relative humidity. They took samples that were stored in the primary package at those conditions, and measured them first at 3 months and then 6 months of exposure. So what you can see here is the reference formulation number 6 maintained dissolution rate above its requirements initially 3 months and 6 months. Formulation number 7 failed dissolution after 3 months, dropping down to 83%, and then continued to get worse. After 6 months, dissolution was only 31% after an hour. Formulation number 1, which contained different excipients, passed at 3 months and failed at 6 months. So this gives you a pretty good idea. There's many other publications showing how increasing water activity can impact dissolution rate. For physical properties, these are tablets. This is some work published by Lee, correlation and prediction of dissolution of benzoyl hydrochloride tablets. Again, the active ingredient itself is not moisture sensitive, but the formulation is. These materials were stressed. By that we mean stored in an open cup for 3 months at 40 degrees centigrade and 75% relative humidity. And what we can see here is the disintegration time went from an initial rapid 2 to 3 minutes up to greater than 10 minutes. And the hardness of the tablet changed dramatically. The thickness and the diameter were largely unchanged. And I know on this slide it's a little bit tough to see the photograph, but there was a dramatic physical, visual difference. And again, if, in our application note, there's more information available on these changes. So just would like to ask another question before we move on. Polling question number 2. So would like to ask the audience a little bit, those of you participating now, there's 152 of you. We've talked about critical quality attributes and how water activity impacts them. I'd love to get your input in terms of where you have concerns, and in this case you can answer yes to as many as, and if appropriate, all Are you concerned that water activity affects growth of microorganisms? Are you worried about loss of potency? By that we mean the concentration of the active ingredient decreasing. Are you worried about the increase in the concentration of impurities, the degradation? Are you concerned about changes in dissolution rate? And do you have concerns about changes in the hardness, friability, or swelling of your compound, your formulations? So again, pretty strong response. We're heading towards 100 people. We'll give it just, there we go, past 100. I'll stop it there. Thank you very much. You can see microorganisms kind of wins the vote, 80, and then pretty similar numbers between potency, impurity. Interestingly, the second highest was changes in dissolution rate, 74%. So thank you very much, everyone, for that. It helps me understand as we move into the next section. So I think it'll become clear that certainly you can test water activity on your incoming materials. It's very valuable. Certainly you can test them during the manufacturing process. But we at Lighthouse think probably the biggest application for water activity testing is going to be looking at how water activity changes over time in the packaged product. So in this section, going to talk about what's the initial water activity, when you first make it. How rapidly does moisture permeate into your package? Milligrams of water per day, moisture vapor transmission rate. And very importantly, as moisture enters the package, how does the water activity of your particular formulation change? So, starting point, it's really impossible to estimate the initial water activity. You must measure it. Here's a pretty good example. These are freshly packaged different pharmaceutical products, over-the-counter products. We had some aspirin, this baby aspirin, entameric coated, 30 ml bottle, heat induction sealed with 1 gram of desiccant package inside. As you might expect with the desiccant in there, water activity of the formulation was quite low, water activity of 0.17. Similar, we had ibuprofen, this time in a 60 ml polyethylene bottle with heat induction seal and no desiccant. Freshly packaged, water activity of 0.52, 52% relative humidity inside the package. Similarly, looking at blister packs, naproxen coated tablet in a blister pack, water activity 0.37, and ibuprofen, a capsule, this case a liquid gel in a blister pack, and of course, as you might expect, water activity of 0.57. Each of your products are going to have a very different initial water activity, and it is essential to know what that activity is as you get into stability studies. It's actually possible to take a moist or high water activity drug, put it into a foil package, which has low moisture transmission rate, but because of the moisture at time 0, you can actually see very high degradation rates of a moist product inside a foil package. So I think the second change I'd just like to show is we've talked a little bit about it. So this is some work where had 8 tablets, gram each, that had been pre-equilibrated at 25 degrees centigrade and 60% relative humidity. So this was done in a desiccator filled with saturated salt solutions. And then I'm going to take those tablets out and put them into a glass vial, and I'm going to add 2 grams of freshly dried desiccant to that vial, stopper cap the vial, and then test it repetitively over time. So, of course, what we all expect is water is going to move from the tablets to the desiccant. I think you would understand a closed system like this, the total amount of water in the system, the total weight of water stays the constant, but water's going from the drug to the desiccant, and the relative humidity inside the package is dropping. So in this particular case, with 8 grams of formulation at 60% relative humidity and 2 grams of desiccant, what we'll see is within 12 hours, we had reached an equilibrium. The relative humidity inside the package decreased to 8% from 60%, and the water activity of the formulation itself and the desiccant, if we took them out and measure them individually, or if we measure them together, the water activity was 0.08. So again, this is showing moisture redistribution. So now I'd like to talk a little bit about moisture vapor transmission rate. Today's presentation is really about USP 922. But those of you that work in packaging engineering groups may be aware that USP has published a new updated version of Chapter 671, Container Performance Testing. So historically, this has been done with desiccants. Take a dry desiccant, put it inside the package, store it at a particular temperature and humidity, and measure weight change over time. As moisture permeates, you gain weight into the desiccant. That's been a very reliable way for testing bottles. It's been done for decades, but the USP committee has recognized that there are significant limitations and measurement errors when using this approach for blisters. So the new updated chapter, which has just been published, the old method I'll call the desiccant method. You're going to put the desiccant inside, and then you're going to put the package into an environmental chamber, 23 degrees centigrade, 75% relative humidity. And among the issues are, is there enough desiccant inside the package to keep this difference, the relative humidity, assumed to be 0 and 75% on the outside, to have accurate values? That delta or driving force must remain constant. And what happens with blisters with limited cavity size, depending on the moisture vapor transmission rate, you can begin to absorb enough moisture where the capacity of the desiccant is being exceeded and the relative humidity inside the blister is increasing, and therefore the moisture vapor transmission rate data is being impacted adversely. So the new method, you simply put water inside the blister. You don't need a lot. 1 drop, a few hundred microliters is fine. And then you're going to take that water-filled blister and put it in an environmental chamber that has lower relative humidity. In this case of the chapter, 25%. And then in this case, you are going to measure weight loss rather than weight gain. So by definition, the humidity inside the blister filled with water is 100%. It's 25% on the outside, and I'm going to measure weight loss. There are some nonlinearities at the beginning of the process as moisture is adsorbing or desorbing from surfaces. So the techniques use linear regression to calculate slope over multiple days. And depending exactly on the blister you're working with, it can take 35 days to collect enough data. So the existing and new methods, this little on the left shows you a linear regression simulated. It's not real data, but just gives you an idea what this would look like. And you're going to take the slope, and that's going to be your moisture vapor transmission rate reported in grams of or milligrams of water per day. But Lighthouse has another way of doing it, and just want to show you. This is what the internal relative humidity would look like if the package was allowed to change over time. So instead of being a static, this is a dynamic or continuous measurement. So different packages have different transmission rates. As moisture enters the package, the relative humidity on the inside is going up, and I will see this nonlinear effect. This is what permeation over time looks like in a real package. So here at Lighthouse, we thought, well, this is interesting. We can actually use this new concept that was shown in USP 671 of taking a blister and putting water in it. But now we're going to take that blister and we're going to treat it like we treat other samples. We're going to put it into a vial, stopper the vial, and we're going to measure the change in the relative humidity or water activity, concentration of water outside of the water-filled blister, but inside the vial. So this is what that data looks like. This is 4 blisters into a vial, and the control samples are blisters that are empty. We can plot this data, we can look at it over time, and instead of doing linear regression, we're going to actually calculate permeation, which is that factor, the exponential term in the permeation equation itself. So this is an experiment that we see many people in packaging engineering and people that are doing accelerated predictive stability work are really quite interested in. There is a high need to really understand permeation rates on blisters. How do they vary between materials, between thicknesses, lot to lot, vial to vial, etc. So this method is very simple. We would be happy to make measurements for you in our lab. We would ask you to prepare samples. It's easy. Take a blister, put some water in it, and then also send us a control without water. We're going to take those and put them into vials, and we're going to hold those vials in a temperature bath, a dry bath, and then we are going to remove them from the bath and test them at time points. We can test very frequently, multiple times a day. We can test over days or weeks or months. And the measurement of the water vapor concentration is extremely sensitive and very precise. We can provide back to you a full permeation curve where the change in water activity was 100% at time 0, and then continue that data until it approaches 0. Probably none of us will continue the experiments until it actually gets to quite a 0 driving force, but carry that out and then report those results back to you. Okay, so this is an example of what's happening with a product inside of a package. So now instead of water-filled or empty packages, I'm looking at this effect. Water is entering by moisture vapor transmission, permeation. What's happening to the drug inside that package? So this is a combination of the nonlinear moisture vapor transmission rate and the nonlinear absorption isotherm for your drug. And what you'll see here is that all these different packages are intended to slow, in most cases not stop, but slow the rate that moisture enters the package. The pill or tablet will absorb water, the water activity of the product, and if there's a desiccant present, and the desiccant will increase. And this process will continue until the water activity of your product equals the relative humidity of the exterior of the package. The rate can be different. It is obviously impacted by the type of package, a bottle versus a blister, the type of blister material, and I hope you understand it's also directly impacted by the moisture absorption therm of your product itself. So not just the package, but the product. So here's another example. There's lots and lots of data published. So this is just a stability study under accelerated conditions, 6 months at 40 degrees C and 75% relative humidity. As usual, of course, there's an open cup inside there. Moisture vapor transmission rate greater than 100 or infinite, whatever you want to say. Obviously, at the end of 6 months, the water activity of the pill, actually after even a day or two, the water activity is 0.75. And these particular products failed They failed by impurity, and they failed by dissolution. They're just not stable for 6 months at those conditions. If I use a PVC blister, it has a little ability to slow down water, not much, and sure enough, the formulation reaches 0.75 within 6 months and also fails. And then there's 3 options here, slightly better packages: a 60 ml bottle, a PVDC blister, and an Ultrex blister. So each of these have lower moisture vapor transmission rates. At the end of 6 months, I have not reached 75%. With the bottle, it's at 0.6, the PVDC 0.4, and the Ultrex 0.2. And it's this primary packaging that now gives me the ability to pass. At 0.6, many drugs will pass, some drugs won't. It just depends. So at Lighthouse, we believe probably the place where you're going to be using water activity the most is during your stability studies. Clearly, you're doing stability studies during the entire life cycle. You're using it during the early days with formulation development and process development. You're using it before you go into the clinic. You're doing stability trials before clinical trials, and you're doing stability studies on a commercial product, filing your NDA or ANDA, and then following your product over time. So we think that there's significant benefits. We know you're doing Carl Fisher testing and total water, HPLC, and other assays. Why add another? If you begin using water activity routinely, you will build an internal relative humidity versus time profile for each product. This is incredibly valuable when you're looking at changes in packaging or formulation over time. You're actually going to be generating a water absorption isotherm for the product if your testing already includes Carl Fisher. So as you're doing these pulls, you're going to know your total water. Now you're going to know the water activity, and you're actually going to generate what does this isotherm look like on a real product over time. As you build these relationships, you will begin to see that water activity is clearly the best correlation to the critical quality attributes, including potency, purity, disintegration, dissolution, hardness, and swelling. And understanding these relationships for your formulation are critical for it to continue to meet its endpoint. And finally, of course, if you're implementing water activity testing, you can reduce the amount of microbiological testing that you need to do during that stability study itself. So I've got another polling question. Launch polling. So this is a little bit about the life cycle and a little bit about the different kinds of departments that you work in. So again, you have the option to say that you're not doing any water activity testing, but would also encourage you, are any of you using accelerated predictive stability, and are you using water activity as part of that? Are you using water activity in lieu of microbiology? Are you using water activity as a part of your formulation development and screening? Are you using water activity in your packaging engineering groups for packaging selection or comparing one type of package to another? Are any of you using water activity during process development? How does the drying conditions or the milling conditions affect water activity? Are any of you currently using water activity in batch release? And are any of you using water activity in post-approval changes? So, looks like the numbers are still going up a little bit, so I will give it a few more seconds. But we still have a fair amount of material to cover, so sorry if I don't give you enough time. Pretty broad reach. I'm very pleased to see that 24, 25, 29% of you, 23%, 30, a lot of people using it in development, a lot of people using it in predictive studies, and fewer of you currently using it in batch release and post-approval. We think with the publication and implementation of USP 922, we're going to see that while it's already being used in development, it's going to be used even more frequently now for batch release and post-approval changes now that it's an approved general method. So thank you for that. Appreciate it. I think the next section is to talk a little bit about how does Lighthouse measure water activity. So that's a photograph of a Lighthouse water activity analyzer. As you're going to see, we use tunable diode lasers to measure the amount of water. It's a spectroscopy technique. We're unique in that we are going to test your samples inside sealed glass vials that are pre-equilibrated. And you'll see that this gives us a couple of significant advantages. We have very high accuracy, typically plus or minus 0.005, 5 times 10 to the minus third water activity units. Unlike other types of instruments, the method is very rapid. We can report this level of accuracy with samples within 5 seconds. And because we're working with pre-equilibrated samples inside sealed vials, we have no issues with moisture memory and no issues with moisture loss. Lighthouse, for its entire history, has been involved in working with the pharmaceutical industry, and our equipment, our methods, our techniques are appropriate for use in CGLP, CGMP. We have a 21 CFR 11 compliant database. We can provide installation qualification and operational qualification, and technical specialists like myself, Paula, and others who come on site to train and help you with validation. So first, just a little schematic. We have a laser diode. It's a very low energy laser. We can control its wavelength extremely precisely, and we tune it to the exact wavelength where water molecules absorb energy at an exact wavelength where nothing else absorbs energy. And then we have a detector where we measure the amount of light that's absorbed. We scan or slowly move this frequency very, very small increments, fractions of a nanometer. So we create a signal that helps us eliminate noise and interference. And the amount of light that is absorbed is directly proportional to the number of water molecules in the headspace. We calibrate with a saturated salt standard. You'll see that. So we know that this number of water molecules equals this water activity. And you'll see that water activity measurements using tunable diode laser, as stated in USP 922, is the only technique that can give you accurate data when your samples contain other volatile compounds. So when you're working with materials that degrade, you're likely going to see alcohols, aldehydes, and organic acids created. These volatile organics play havoc with traditional water activity systems and have zero impact on our ability to accurately measure with laser spectroscopy. So our sample preparation technique is very simple. You're going to take your primary package, be it a bottle or a tablet. You're simply going to open it. You're going to put that pill, capsule, tablet, powder, whatever form it is, into a glass vial, put in a stopper and crimp on a cap. And you're then going to put that sealed vial into a temperature control. It could be your environmental chamber, it can be a dry bath, or it can be a temperature block. There's some photographs here. You can have as little as 10 milligrams of sample. You can have as much as 25 grams. And you're going to let them come to equilibrium before you measure them in our instrument. Instrument's simple. There's a cavity or an opening at the top. You simply take the vial, put it into the instrument, press the measure button, and the instrument, as you saw on the diagram of the optics, includes a very precise temperature controller where this vial resides inside the instrument. So we will report both the water activity and the temperature of the sample. And we can, because it's been pre-equilibrated in a sealed vial, we can make a very accurate measurement within 5 seconds. So this is a picture of two different saturated salts, USP 922, and really all the instruments, any type of water activity is typically calibrated with a saturated salt. For those of you that have not seen them before, they really look like a slurry, don't they? You can see the salt crystals in the bottom. Of course, the liquid above it is therefore saturated. The one on the vial on the left is sodium chloride. The bottle on the right is lithium chloride. And again, when you think about water activity, the chemical potential, how tightly is water bound? Saturated sodium chloride has a water activity of 0.75. Lithium chloride is dramatically different, has a water activity of 0.116. It very strongly binds water. This data is summarized in chapter 922, and almost everybody references Greenspan, publication 1977, with a long list of many, many, many saturated salts and their dependence on temperature. So our instruments have we call it a GXP mode. The supervisor or the group leader is going to write a method. The method can include a linearity check. So this is what the output looks like. Here the operator has tested 5 different saturated salt standards. They all fall within they're plotted along a linear line, and they all fall within a very narrow band of accuracy, expressed as the red lines. Because it's been written into the GXP method, the system must pass this test before you can move on and test samples. And this is what that data looks like. This is a little bit like an intermediate precision study. So this is 6 different saturated salts tested on the same instrument, but tested with 5 reps of each over 5 different days. So that's 300 data points appearing on that line, and you can see it's an extremely straight line with almost no scatter, a slope of 1.0064, and an RSQ of 0.9992. The instrument performance over this week is really quite impressive. So I mentioned earlier that we test inside sealed vials, and I think this is just something I'd like to spend just a couple of minutes on. It's about the robustness and the throughput of the instrument. So the time required for a sample to reach equilibrium is typically much, much, much longer than the time that's required to make an accurate measurement. Moisture must first desorb from your tablet or capsule and must reach equilibrium with the air in which it's going to be measured. Please remember, water activity is this exact measurement of this exact equilibrium. If it's not at equilibrium, you are not seeing water activity. So when samples are tested in a sealed vial, we can separate the time required to reach equilibrium from the time required to make the measurement. They're 2 completely independent things. We can therefore report water activity only after the sample is known to be at equilibrium. And because we're inside a sealed vial, we can isolate and protect the headspace from any moisture loss or moisture gain, either from the environment or in the case of other instruments because of inside the measurement chamber itself. So I'd like to show some data that supports that. This is just some aspirin tablets. We drop them into a vial and seal it, and we're going to test it repetitively over time. So I'm showing water activity initial, and then after 2, 4, 6, 8, 10, 20, 30, and 40 minutes. Aspirin actually comes to equilibrium pretty quickly. So what I'm showing is the water activity goes from 0.274 down and ultimately reaches equilibrium at 0.174. If you had that sample and you waited only 4 minutes, you would measure water activity and it would give you 0.1197. You would have an error of 14% from its true value. As you wait, if you're patient enough, as you wait, you will get closer and closer to the true value. After 30 minutes, we were within 0.001 AU. And this is really representative of pills and tablets that come to equilibrium quickly. We've seen many formulations that can take 3 hours for this to occur. So other thing that happens with a sealed vial is I don't have to worry. Traditional instruments, be it a chilled mirror or a resistance capacitance hydrometer, my sample is going to be in a plastic cup. I can have a cap on the cup when it's outside the instrument, but I have to remove the cap and take that cup, place it into a chamber, lower the lid, and seal the open cup inside the chilled mirror or capacitance measurement chamber. So this is a paper that was published from some scientists at Merck in PharmTech back in October of 2005. So these are preconditioned tablets that were sample 1 preconditioned at 20% relative humidity, sample 2 preconditioned at 85% relative humidity. And just a really simple and kind of obvious experiment. Can I test a dry sample, then test a wet sample, then test a dry sample again, and what happens? USP 922 warns you that this is part of a robustness test and things that need to be carefully documented. So in the case of the Lighthouse instrument, we're in a sealed, testing in a sealed vial. There is no memory. You can make these measurements as quickly as you want between wet and dry or in any order. But on the systems that use open cups, I can see some pretty large memory. I can see water activity gains of 0.015 to 0.057. Relatively speaking, those are measurement errors of 7.5 to 28.5%, and obviously has a pretty dramatic impact on your ability to interpret data. So I thought I'd just spend a minute and say, why are these things so sensitive? If that cup contained water, we would have a relative humidity of 100%, a water activity of 1.00. Well, how much water is that? So at 25 degrees centigrade, the partial pressure of water is 32 millibar, and that corresponds to a concentration of 19 micrograms of water per cc. If I'm at 30 degrees centigrade, it's 25 micrograms of water per cc. And if I'm at 40 degrees centigrade, it's 44 micrograms of water per cc. When we're testing inside a sealed vial, you can see that while I have to reach equilibrium, very little water is going to leave your tablet to go into the headspace. There's going to be a very, very small change, and that trace microgram quantities of water is going to be trapped inside that vial. If I'm, on the other hand, working with a system where I have to open the cup, I'm going to have a lot of issues. So very trace levels of water, if they're left anywhere inside that measurement chamber, can have a dramatic impact. That's what the memory is. You can see here a few micrograms of water left inside is going to have a big impact. In addition, if I'm storing things at elevated temperature and waiting for them to come to equilibrium, very, very small changes, if any of that water migrates out of that chamber, it's going to have a dramatic impact on the measurement also. So in the case of Lighthouse, working with sealed vials, samples can be held at elevated temperature for hours, days, and weeks with no measurable change, either in water activity or the weight of water. So in conclusion, USP 922 will become an official chapter implemented May the 1st, 2021. Water activity is very useful and very different than Carl Fisher. It measures the free and loosely bound water in your oral solid dosage forms, and it correlates quite effectively to chemical and physical changes that were important to you. The Lighthouse Water Activity Analyzer is unique. It uses tunable diode laser measurement technique, and it's the only instrument that does testing inside sealed glass vials. It will therefore give you extremely accurate and precise data in as little as 5 seconds. It meets all requirements of CGLP, CGMP, 21 CFR 11, and obviously the requirements of 922. And we have a whole team of people that can help you, including post-COVID, particularly travel to your site to do on-site validation work with you and training. So if you don't mind, before I answer your questions, one last question here. We're really quite interested. For those of you that are doing water activity testing, could you give us an idea of what type of technique you're using? These are all the techniques that are described in USP 922. Chilled mirror, I'm going to make the guess, is the most common. Let's see if I'm right. A capacitance hydrometer, a resistance hygrometer, a tunable diode laser spectroscopy. Some of you, particularly those of you that are working with accelerated predictive stability and other fundamental R&D functions, may be using dynamic vapor absorption. We didn't cover that in today's talk, but I'm interested if you're using it. And then, of course, some of you have no water activity testing. So, well, I'm going to lose the bet that it's chilled mirror. Looks like tunable diode spectroscopies and dynamic vapor absorption are coming out the highest. That's very interesting. But we're going to give you just a little longer. Am I running out of time? I've run out of time. Paula, you want to take over for a moment?
Yes, I'm here busy answering some questions already from the Q&A, but yeah, I do have a couple for you, Mike, which I have kept, so you can also discuss a bit. You let me know when you're ready for it.
I'm ready.
Okay. So I have one from the rather beginning of the presentation. The question is, there is any reference of water activity levels for different ingredients? If it results in a high water activity through the analysis, what do we do? What do we need to do?
That is a great question. Many of the pharmaceutical ingredient manufacturers have already begun to publish water activity data on their individual components. And you will see that the ingredient companies have some really valuable information about how to interpret that information. So it turns out that the compounds that have high water activity may be the most useful ingredient for protecting your formulation. The ingredient manufacturers themselves have already been or are beginning to provide water activity data, both for the kind of compound they make. How about my starch? And in some cases, we're actually seeing that they're now providing this on the batch release for their individual batches of those components, because water activity is known to be so important. So it's important to know what it is, but when you ask the next question, do you need to be worried about that? That's where I would encourage you to work with us at Lighthouse and with other experts in water activity, because a high water activity ingredient may actually protect your formulation, as surprising as that sounds.
Good. I do have another question, and I believe it's regarding the part that you were talking about, the moisture vapor transmission rate studies. What's your limitation in regards to blister card size to be able to put into a vial?
Right now, we would like to keep that to 1 to a few blisters. We have not done any work yet with the somewhat obvious question you would ask, what about 10 blisters on a card, which is kind of a common number. We'd love to have a meeting and a discussion with you. There are some ways that we perceive that we can do that. We haven't done that yet. So we would, one of the ways to think of it is we're going to use a vial that is got an internal volume of 50 or 100 milliliters, a large neck size at the top, and then put the blisters inside. As we want to go to cards, we're going to have to use some custom glassware, and we haven't done that yet, but we'd love to be able to discuss it with you.
Good. Is a special stopper required for the sample preparation that you were describing? So the stopper moisture does not impact the water activity inside the vial?
It's a great question. Lighthouse does a tremendous amount of work with injectable pharmaceuticals, and we use standard injectable pharmaceutical vials and stoppers and caps. It is not necessary to extremely carefully control the water activity or residual moisture in the stopper itself. It is clearly possible to do that. Our scientists and our application labs have been looking at the difference between stoppers equilibrated at different relative humidities and dried, and we'd be very happy to have that discussion with you. But we have found little to no difference using traditional chloro or butyl rubber stoppers that are used most commonly for injectable pharmaceutical products.
Thank you, Mike. Perhaps one more question. What is the critical impact of the water activity measurement procedure using the Lighthouse instruments once the USP 922 chapter is effective?
That question may be coming from a current Lighthouse user. So every Lighthouse water activity unit from serial number 200 and up, Lighthouse has been delivering these systems since the year 2010. Every one of the units with serial number 200 and up has either already or can be upgraded to include our latest version of software and our 21 CFR 11 compliant database. Our team is capable of writing and executing IQs, OQs, and PQs on any instrument that is already installed. So if I understood your question correctly, if you currently have a Lighthouse instrument, it meets all the requirements of USP 922. If you wanted to meet all the other requirements for GLP and GMP, your system, depending on how it's configured with software, is either already there or can be easily upgraded, and we'd be happy to talk to you about those upgrades.
Good. Thank you, Mike.
All right, I think I'm gone a little bit over time. We are going to send each of you a link to the recording of this webinar. We'll send it to everyone who pre-registered, everyone who attended, and everyone who pre-registered. Some of you are going to have an interest in the Lighthouse Water Activity Analyzer. So a week or so after, or 2 weeks after the seminar, we're going to be sending a link to a video of our team operating the instrument and showing you some of its properties. We are very happy to schedule meetings with you, with any of our technical support